Welcome. Today we are going to discuss perhaps the most difficult topic that we have introduced in this course, and that is the rate of a chemical reaction as we understand it by using the principles of molecular thermodynamics. Now, as chemists, we always come across chemical reactions, but the view that we take of a chemical reaction is usually a macroscopic view. So, when we go to the laboratory, we do see chemical reactions where you can see a variety of effects like the ones that have been highlighted over here. There may be a large amount of heat released, there may be a drastic change in color accompanying the chemical change that you are observing through the chemical through your experiment. Now, if I want to find out the rate of such chemical reaction, in that case the standard thing to do schematically is you write down a stoichiometric chemical equation as shown here, and then you write the rate of the reaction as the rate at which the reactant A is degrading in time, so that the products P and Q are formed. And in general, what we find is in the experiments, it is found convenient to study if the rate depends on the concentration of the reactant, which means that whether it is directly proportional to the concentration of the reactant A, or it is proportional to the square of the concentration of the reactant A, and so on and so forth. So, this quantity n, this is traditionally known as the order of the reaction. And then what we do in the experiments is as follows. We try to, for example, plot the change in concentration of A as a function of time, and depending on what the order of the reaction is, you can have for the first order reaction a rapid decay in the concentration of A as it converts to P and Q. But if I have a second order reaction, then the decay profile of A, concentration of A in time is going to be different. So, then from this experimental data, what one does is one finds out the slope of this curve and fits it to this equation and gives us the rate of the reaction. Now, in this rate of the reaction, the important quantity is this rate constant, which is a proportionality constant that tells me that for the given reaction, how the rate will be estimated for a given concentration of A. So, the challenge of today's lecture is if we can give a microscopic interpretation of this rate constant K, because as you see here, the rate that we are measuring, we are using all the macroscopic equipments to do that. Concentration of A is something like moles per liter, which once again is a macroscopic concentration. Therefore, K is also a macroscopic quantity, and we are set to find out a microscopic interpretation of that. Now, in order to do this, obviously, I need to set up the microscopic view of the chemical reactions. And when I do this, we understand that as chemists, we are usually familiar with a molecular picture like this associated with a chemical reaction. For example, here you see that this molecule appears, approaches this molecule and takes one atom from it and leaves. On the other hand, in this particular case, as you see that the green molecule comes in and takes away a group like this is an NH3, which was probably coordinated to the yellow atom, and takes it away. Or you can even have very complicated situations, for example, in an enzyme catalyzed situation. So, this is a proton transfer reaction within an enzyme, where you see that these water molecules are exchanging one proton between themselves, so that one water molecule loses a proton, and eventually this proton lands up on this particular nitrogen atom. So, the question is, as you see that the system is undergoing a series of transformations, whereby it goes through a very large number of microscopic states. Therefore, if you want to find out that macroscopic rate constant, you need to take into account all these microscopic states that are available to the system. Therefore, if we ask this question, how do we map the macroscopic rate constant to the molecular model that we are usually using to understand them? The answer is we need to average over all possible microscopic states of the chemical reaction. And therefore, this brings us to the question of how do I model the microscopic states in a system where, unlike the previous cases that we discussed, we do not have non-interacting molecules. Right now, I have interacting molecules which participate in the making and the breaking of chemical bonds. And therefore, you understand that this problem has taken an enormously difficult proportion. And in this case, with the development of quantum chemistry, we now know that the different microscopic states of these interacting molecules are represented in terms of what is known as a potential energy surface. So, what is the potential energy surface? Typically, a potential energy surface in terms of two variables, say x1 and x2, they would look like this. Okay? And now you see that this potential energy surface has a very complicated structure. It has some kind of what is called a local minimum, where the potential energy takes up the minimum value for certain values of x1 and x2. There may be other local minima like one here, but there can be global minimum where the potential energy of the system, of this interacting system, is the minimum amongst all the values of x1 and x2 available to it. Now, a chemical reaction is observed or is modeled as a transition from one of these minimum by climbing this hill and going over to the minimum on the other side of the potential energy surface. Therefore, this top of the hill, which allows us to go from one side of the hill to the other, is known as the saddle point, which is the location of the so-called transition state of the reaction. Now, we are going to use very simple model systems to understand how such structures appear and how to interpret them, so that we understand the task at hand, that is the averaging over different microscopic states associated with the chemical reaction. So, for this purpose, let me once again show you that, well, the task is, I have a picture like this, where I can have something called a reaction coordinate that distinguishes between the reactant and the product state, and there is an energy barrier in between, and therefore, I would like to find out the energy surface such that the transition state is located at the maximum as the system passes from one minimum to the other minimum as the reaction progresses from the reactant to the product state. Now, if I go ahead and try to ask this question, how do we obtain such potential energy surfaces? So, let us go back to a very qualitative consideration of the quantum mechanical model of such interacting systems. So, let us say that I have m nuclei located at the positions R, capital R alpha, and with charges and masses and momenta specified for each of them. I can also have capital N electrons at their locations for a given microscopic state with their respective charges, masses and momenta. And then the picture for a system like this is given here. So, can you identify what kind of molecule is this? Obviously, this is H2 plus, where I have one hydrogen atom at the position, one hydrogen nucleus at the position capital R1. I have another hydrogen nucleus at the position R2, and there is a single electron which is located somewhere here with a position vector small r1. Okay. So, if I now set up the Schrodinger equation for a system like this, what I would get is I have to start from the Hamiltonian of the molecule. So, the Hamiltonian of course now requires many different terms to describe the contribution of kinetic energy as well as the potential energy of the different things present in the system. So, initially I have this term which represents the Hamiltonian corresponding to the kinetic energy of the capital M nuclei present in the system. Then I also have the part of the Hamiltonian which represents the kinetic energy of all the capital N electrons in the system, and then I start having the potential energy term. So, this is the potential energy term for the interaction between the different nuclei present in the system, and this is the term where you have the interaction between the different electrons present in the system. And finally, I have one term which represents the interaction between the nucleus of having the charge Z alpha into E with an electron having the charge minus E, and they are located at these two positions. Now, a simplified and compact notation of the Schrodinger equation to be solved then is given like this, where these red capital X's represent all the position coordinates of all the nuclei present in the system, and these blue small x's represent the coordinates, position coordinates of all the electrons present in the system. Now, under the Born-Oppenheimer approximation, we know that the motion of the electrons and the nuclei, they take place at very different time scales, as a result of which we can actually solve the Schrodinger equation for the electrons by keeping the nuclei fixed at certain positions. So, this is exactly what we have done next, and we find that now the Schrodinger equation that I have to solve is given by an equation like this, where capital X, these positions are fixed. Now, when we solve these Schrodinger equations for a fixed value of x, the results are something like this. So, by solving the Schrodinger equation, we get the energy eigenstates. So, let me say that if I fix the this is a particular molecule that I am looking at. So, it has one atom which is a red atom, another atom which is a blue atom, and the center to center distance is given by capital R. Now, if I fix it at this particular value of R, which corresponds to this value of R here, this is the ground electronic energy obtained by solving the Schrodinger equation, and this is the first excited electronic energy that is obtained from the solving the same equation. Now, as you understand that I can fix this relative separation of the two atoms at different values. For example, if I fix it at this value, which is smaller than this before, then let us say that this is the corresponding value of R, and these are the corresponding values of the ground and the first excited electronic state of this molecule. Now, this way we can actually generate all such energy values for different interatomic separation within the molecule, and as you understand that if the two atoms invade each other's space, then obviously the energy is going to be very high, and we have not shown it in this picture. So, by solving the Schrodinger equation, what is it that we are getting now? What we are getting is, we are now joining all the ground state electronic energies for the different geometries through this dark blue line, and all the first excited state electronic energies with this red line. So, the lower curve gives me the potential energy curve for this particular system, and this is the ground state potential energy curve. And this is the first excited state potential energy curve obtained by solving the Schrodinger equation and the first excited state solution of it at different geometries. So, this is possibly the simplest representation of a potential energy between within a molecule when I am changing their relative geometry. Now, if I have a more complicated polyatomic molecule, you understand that in addition to R, I need to have many more parameters, and in that case the potential energy curve does not remain a one dimensional two dimensional curve, but in this case what I will get is a molecular potential energy hypersurface with a very large number of dimensions in the case of a complicated molecular system. Now, what is the use of this molecular potential energy surface? As you see, if I write down the nuclear part of the Schrodinger equation, I find that this electronic energy actually plays the role of the potential energy on which the nuclei move. Okay? And therefore, here comes the importance of the electronic energies for fixed geometry of nuclear, fixed geometry of the nuclei, because the solution of the electronic Schrodinger equation gives me the potential energy for nuclear motion. But please remember, there is no temperature in Schrodinger equation. Therefore, whatever I have solved here, it has been calculated at T equal to 0 Kelvin. So, in Schrodinger equation, we are solving for the microscopic value of the microscopic description of the system at T equal to 0 Kelvin. Therefore, the potential energy surface that I get is a valid description of the interaction between the different parts of the molecules at 0 Kelvin. Now, then obviously, the question is, at a finite temperature, how do I represent the chemical reaction on the potential energy surface? This is in general visualized as a rolling of a ball on the potential energy surface that I will show to you later. So, depending on what the amount of thermal energy available to the system, then the kinetic energy is high or low, and that ensures the extent to which the system can explore different geometries. If you are at a very low temperature, in the previous case, the two atoms cannot have large kinetic energy, and therefore, they will be forced to stay in close to each other, thereby sampling only the geometries near to the minimum of the potential energy surface. Now, let me take an example. So, this is a chemical reaction which is very simple, a hydrogen molecule interacting with a hydrogen atom. So, initially hydrogen atom A and B are bonded to each other, and there is another atom C which is coming close, and at the end of the reaction HA is isolated and a bond has been formed between HB and HC. Now, if I look at the potential energy surface that I get from here, this is described in terms of two distances. One is the distance between the hydrogen atoms A and B, and another one is the distance between hydrogen atoms B and C. So, as you see that in the reactant state A and B, these two must be having a minimum potential energy value, indicating that there is a stable hydrogen bond, stable chemical bond between them, and therefore, I see that this is where the reactant state for this reaction must be located, and under that condition I must be having a large distance between B and C. Okay. So, here I have this RBC, it is going like this. So, since RBC increases in this direction, therefore, at this point RAB is small and RBC, that is the distance between the hydrogen atoms B and C, that is very large. Now, as the system goes to this state where there is a chemical bond between B and C, what do I expect? I expect a minimum in the potential energy corresponding to a small distance between B and C. So, that exactly appears over here, and under such circumstances I must be having a large distance between the hydrogen atoms A and B. So, as you see, as expected, this minimum appears at a very large value of RAB, and here as you see that we have connected equal energy points by these blue curves. Okay. Now, this is a three-dimensional picture. Most of the times it is found that it is much more easier to look at what is known as a contour diagram of this potential energy surface. So, in this potential energy surface contour diagram, as you see, each minimum corresponds to this kind of contours with very small diameter. So, this is a minimum which is corresponding to this product state. This is the minimum which corresponds to an equilibrium bond length between the hydrogen atoms A and B. Now, these blue lines now appear as these contours or curves in the two-dimensional space, and here you see that larger the diameter of the contour from some kind of a center, which means that you are looking at higher energies. Therefore, this energy would appear here, but this energy would appear at a much lower value like this contour or this contour. Okay. Then the question is, what happens when the reaction takes place? When the reaction takes place, the system, for example, starts from the reactant well, potential energy well from here, then climbs up the energy hills. So, if you come from this region to this region, you are going from lower energy to higher energy. Then you pass through the saddle point and then you decay to the product state. But not always that the system, when it attempts to scale this mountain of potential energy barrier, it will be able to do it just as cleanly as this. So, it gets it climbs up this way and then it goes this way and tries to find its path through the saddle point and decays like this. And many of the cases, it so happens that the system is unable to climb the barrier at all, because of the very high energy required for this. Now, if it so happens that at a given temperature the kinetic energy available to the system is not enough, then it attempts to scale the energy barrier, but once again falls back. And while doing this, it may fall back directly like this, or it may try to rattle in that particular potential energy well and eventually go back to the minimum. Now, once it is near this saddle point, so the saddle point is this region. Now, once it is near this saddle point, it has two options. The first option that it has is, okay, I will go from this lower energy value through this direction, through the saddle point and come here. So, that is one kind of option it has, or while it reaches the saddle point, instead of going here, it can try climbing in other directions like this or this. But as you see that the second direction requires a very large amount of energy, and a system, as we know, that is always trying to minimize the energy cost because it has limited thermal energy available to it. As a result of this, I would say that in the plane RAB, RBC, there will be only some combinations of RAB and RBC that will allow the system to pass from the reactant well to the product well. And those combinations of RAB and RBC, which take the system from the reactant to the product well through a minimum energy path, is called the reaction coordinate. So, if the system moves along the reaction coordinate, it undergoes a transition from the reactant to the product state through a minimum energy path. Okay. And in this particular case, the combination of RAB and RBC that gives you this minimum energy path is known as the reaction coordinate. And therefore, once we have established what the reaction coordinate is, then we can project the potential energy surface along this reaction coordinate to get this very familiar one-dimensional potential energy profile that tells me that, well, I have here a potential energy well corresponding to the reactant, where there is a stable chemical bond between the hydrogen atoms A and B. There is another reactant well over here, where there is a stable chemical bond between the hydrogen atoms B and C. And during the transition from this state to this state, the system had to pass through a transition state here, where A is still bonded to B, but it has started forming its bond to C. So, this is the hypothesis of a transition state or an activated complex. Now, within this molecular picture, then one tries to understand what happens when I adopt such a picture for the molecular model of the chemical reaction. So, as you see, as the system samples many microscopic states accessible to it during the reaction, this is how the chemical reaction is represented as a motion of a ball along the reaction coordinate, starting from the reactant well and climbing the activation barrier, and then finally relaxing to the product well. So, this is the molecular picture, and the many different ways that the system can do this is the array of molecular thermodynamics. So, let us now quickly discuss how to get the rate of chemical reaction from the perspective of molecular thermodynamics. And in this context, we are going to use the same potential energy profile along the reaction coordinate, and we are going to consider this very simple kinetic scheme, which is A and B reacting with each other, and while doing so they rapidly establish an equilibrium to produce the transition state or the activated complex X dagger, which subsequently decays into the product P. Therefore, in this picture what I have is this minimum corresponds to the two reactants A and B, and then this maximum corresponds to the formation of the activated complex X dagger, and finally, the second minimum in this profile corresponds to the product or the products. Here there is only one product P. So, once we have established this kind of a picture, in that case the rate of the chemical reaction can be given by this rate constant multiplied by the concentration of A into concentration of B. But please remember, concentration of A is given in terms of moles per liters. So, all the quantities that I see here are macroscopic quantities, but the picture that I am associating here is a microscopic one. So, what I am going to do is I am going to write down according to this kinetic scheme valid at the molecular level, what can be V? V is going to be proportional to the concentration of the activated complex, and the V is therefore given by K dagger into the concentration of the activated complex. So, K dagger is the rate constant with which X dagger decays into the product. Now, with this we can also write down, if there is an equilibrium between A and B, the molecules of A and molecules of B forming with the molecules of the activated complex, this can be written down in terms of partial pressures like this. So, I am now assuming that this is a gas phase reaction where the total system is maintained at a given temperature T and at a standard pressure P naught. Okay. So, under such condition I can also say that, well, since all my rate laws are written in terms of the concentration of the species, I will replace the partial pressures in terms of molar concentrations. And if I do that, then I can write down the equilibrium constant for the formation of the activated complex like this. So, this is done in terms of things that we know very well from our initial knowledge of physical chemistry. Now, we will go further and then say that, well, from the equilibrium constant I can rewrite the concentration of the activated complex, and now combining these three equations I can write down K reaction is a quantity which depends on this constant K dagger and the equilibrium constant K equilibrium dagger. So, the task of molecular thermodynamics is evaluation of this rate constant small k dagger and this equilibrium constant K equilibrium dagger given the scheme as shown here. Now, this is something for which we can use what we have learned before for chemical equilibrium between ideal gases. So, this is given in terms of an expression like this. We have seen this before, but the difference here is now the different components of the reaction, these are present at different energies, and that is the reason why this additional exponential term appears. So, what are these E naught and Qj m naught? So, what I find here is EA, that is the activation energy, as shown here. This is nothing but the E naught that I have shown here, and that is the 0 Kelvin potential energy of the activated complex minus the same energy for the reactant A minus the same energy from the reactant B. Therefore, if I know this potential energy profile from the solution of Schrodinger equation, I can find out EA. At the same time, I see that I am using these QJ terms. For each component in the reaction mixture, this is nothing but the molar canonical partition function for the jth component at a given temperature T, where the total volume of the gas corresponds to the standard pressure P. So, using these two notations, what we can do is we can rewrite the K equilibrium like this, and now I understand that I can further simplify this algebraic equation and have an expression of Q equilibrium in terms of this E naught, then Avogadro number N A, and the molar partition functions for the three components in the equilibrium mixture X dagger, A and B. Now, let us assume that the difference the system, when it forms this activated complex, there is it has several bonds in it, and one of these bonds is executing a very loose vibration. Okay, and this loose vibration, it is so weak that it leads to the decomposition of that particular bond. If that happens, it is very easy to say that X dagger, which may be comprised of many atoms, but one vibrational mode of that atom is loose, and that is becoming loose as the system that is loose, and that bond is broken as the system goes from the reactant to the product state across the transition state. As a result, I can write that this Q naught of X dagger, this is Q loose, that is the vibrational partition function corresponding to the loose vibrational mode, multiplied by the vibrational partition function for the rest of the vibrational modes for the complex X dagger, where because of strong bonds present within X dagger, they are not undergoing any loose vibration. Now, a loose vibration means a very small value of the intrinsic angular frequency of vibration, and under such condition it can be very easily shown that Q loose is going to be given by this particular expression, it is kBT by h nu dagger. And therefore, under such assumption, I say that I am actually able to find out what the molar partition function, standard molar partition function for the activated complex is in terms of a loose vibrational mode. Now, under such condition, then I know how to write down the equilibrium constant, and it is this angular frequency with which X dagger would decay into the product. Therefore, K dagger is also a nu dagger. And this is of course valid if the system, once it reaches the transition state, immediately decays to the product and never comes back to the reactant state. Now, if I combine these two conditions, then what I get is a molecular expression for the rate constant K reaction in terms of the molecular quantities like Q bar X dagger, Q AM naught, Q BM naught, etc., etc., and this is the Eyring equation. So, what we have learned from here is the K reaction can indeed be mapped onto the experimentally known result of Arrhenius equation. We have retrieved this exponential term which includes the activation barrier, and we have a pre-exponential term that depends on the molecular nature of the participants in the reaction. So, to conclude, first of all, in order to understand the microscopic states of the reaction, we have introduced a one-dimensional potential energy profile for the reaction. Then we realize that for a certain amount of kinetic energy available to the system, with the thermal energy available to the system, it is most probable that most of the system reactant molecules will remain here executing small amplitude motion within the reactant well. Now, what happens is it is much low, the system having a high potential energy and scaling this barrier is a less probable process, because the probability of a system being found here is e to the power of minus beta e, and therefore, with increasing value of e, the probability decreases. So, what happens is, with some accessible amount of kinetic energy, the system tries to climb up and then it comes back again. Okay? And then finally, under certain conditions, very rarely, what happens is a trajectory, a system can start from the reactant well, and then according to transition state theory, it crosses the barrier and eventually it settles in the product well. Therefore, if I want to calculate the rate constant for this kind of a reaction, I must take into account what this potential energy profile is, what are the mechanisms through which the system can climb the barrier, cross the barrier and then settle into the product, and all such barrier crossing events needs to be taken into account for the averaging to calculate the rate constant of the reaction. Thank you.